doctors of medicine. Today we continue with our embryology playlist. So the main focus for today's video is to talk about the development of their hearts. So before we get into the interview details, let's take you back to our 3-week-old embryo. So we are looking at our embryo from above. So here you can see this embryo, right? We need to see this layer of cardiogen, this layer that we refer to as the cardiogenic area. So what happens is between day number 16 to day number 18, we have cardiogenic cells that migrate from the epiblast and lie into the region of the splanchnic mesoderm. But they lie in that region in the cranial aspect. If you can see, this is the cranial aspect in a horseshoe structure. And we refer to these cells as our primary heart field. So this primary heart field is going to be responsible for, number one, the development of our ventricles, primitive ventricles, and primitive arteries. Right? So what also happens is that between day number 20 and day number 21, we have formation of our secondary heart field. So now, this secondary heart field is going to be responsible for, number one, forming a small part of the ventricle, also forming the outflow tract, also forming the truncus arteriosus. Right. So this is just to show you that. So from this, we want to look at an embryo that's undergoing folding, as we discussed in formation of body cavities. If you look right here, we can see that this embryo is undergoing lateral folding, but of particular importance for this video are these two tubes. So these androgenic cells that we mentioned here, they are going to form a tube. And that tube is going to undergo a canalization period. When it undergoes that canalization period, that's when it becomes a heart tube. So we have got these two endocardial tubes over here. So we need to pay particular attention to what happens in lateral pre-folding. So as we continue with our lateral pre-folding, we realize that these two tubes are going to fuse. Right? When they fuse, they are going to form this heart tube. Right? So when we form our heart tube, just a brief of the history of this heart tube. The inner cell lining, we refer to it as the endocardium. Right? So outside the endocardium, we have got this red layer, which we refer to as the myocardium. So this myocardium is the one that's responsible for secreting that gel that surrounds is responsible for secreting that gel in the heart that we refer to as the cardiac jelly. Right? So we now have two layers. Right? We also need to know that from the septum transversum, which is now in its original position, we are going to see that we form what we call proepicardial cells. Right? So these proepicardial cells, they're going to move, they're going to go over the sinus venosus, they're going to surround the heart. When they surround the heart, we refer to them as the epicardium. Right? So this epicardium, we can also refer to it as the visceral pericardium. Right? And that layer is the one that's going to contain our coronary arteries and nerves. Right? So now we've talked about the embryology. Right? We also need to know that this tube is suspended in that tube. Remember, we need to talk about the intraembryonic cilium, but now we are calling it the pericardial cavity because it contains the heart. So it's attached to the dorsal aspect by something, a piece of mesoderm that we refer to as the dorsal mesocardium. So it's suspended by the dorsal mesocardium. Right? So particular importance of this dorsal mesocardium, we need to know that when it degenerates, we are going to form what we refer to as the transverse pericardial sinus. Okay. Now that we're done talking about this, we want to look at these heart tubes, but in a different way, because this is a sort of a transverse section. Now we have to look at it from above. if we look, we have removed everything, we are just now left with our endocardial tubes. We realize how these two tubes, how they move closer together during lateral folding. They keep on moving, and then you realize that there's this diagram showing fusion of our primitive heart tube until we have formation of this beautiful looking heart tube. Right? What we also need to know is when we discuss body guard, we said that we form, so we now have three tubes. We have the neural tube. And when we spoke about body cavities, we formed the gut tube. Now, in front of that gut tube, this is where we are going to form the heart tube. So we now have three tubes if we look at the cross section. Now, we want to talk about a bit of anatomy relating to this heart tube. Okay. Now, we want to look at the anatomy of our heart tube. Now, if we look at our heart tube, we have sort of zoomed in and we have expanded it. From here, we see that the heart tube has various number of divisions. So we start from the bottom going up. So we call this area below it, we refer to it as the sinus venosus. And then just above the sinus venosus, we refer to that layer as the primitive atria. Above it, we have the primitive ventricle. Above it, we have the proximal third of the bulbous cordis. Above it, we have the conus cordis, or which is also the middle third of the bulbous cordis. And we have the distal third of the bulbous cordis, which we also refer to as the truncus arteriosus. So from this, it's kind of difficult to imagine how this straight tube will look like your complex heart. So from this one, what you need to know is how blood is moving through this heart tube. So we need to know that blood is coming into the sinus venosus through the right and left wall, through three different kinds of veins. We have our number one, the umbilical veins. Number two, the common cardinal veins. Number three, the vitelline veins. Right? So what they're doing is they are bringing the blood into the heart. So when the blood enters the sinus venosus, it moves through the primitive atrial, the primitive ventricle, the bulbous cordis, and then you realize that it moves out through the outflow tract. Okay. So this is how circulation is distribution in the embryo. So why we are interested in these structures is because these structures are the ones that have a significance, because they are going to contribute to various structures in the normal human heart that we know. Okay. So we have what you call, if we look at the sinus venosus, it is the right and the left one. So from the left one, it will degenerate those three veins. And that left one will form two veins. Right? So it's going to form number one, the oblique vein of the left atrium. Number two, the coronary sinus. Right? And also, if we look at the right one, we have the right common cardinal vein, which will form the superior vena cava. And we also have the right vitelline vein, which is going to form our inferior vena cava. Right. So from there, we move on to the primitive. Also pointing out that this sinus venosus is going to end up, the right one is going to end up being incorporated into the right atrium. And it's going to form the smooth part of the right atrium. Right. We move on. We have got the primitive atrium. Right. Since the sinus venosus is going to form the smooth part of the right atrium, we also have a trabeculated part, which you refer to as the muscular pectinate. That trabeculated part is going to come from the primitive atria. So the primitive atria is going to form the rough parts of both the atria. Now, the question, what about the smooth part of the left atria? So what we need to know that when we are undergoing septation, just lateral to the septum primum, we form our initial pulmonary vein. So This initial pulmonary vein is going to form around the time while we are forming our lung buds, and it's going to extend from the heart to the lung buds. We need to know that when these events are not happening in steps, but they're happening at the same time. So while we are folding our heart, the lung buds are forming, the pulmonary vein is entering the heart, at the same time it's going upstairs to connect with our lung buds, right? So though that pulmonary vein, as they increase in number, they are going to form the smooth part of the left atrium. Very good. So we now move from this. We move to the primitive ventricle. Right? So the primitive ventricle is the one that's going to be responsible for formation of the rough part of the left ventricle. What about the right? So we also, like we were saying, the bulbous cortex is divided into three: the truncus atrius, the conus cordis, and the proximal part. Right? So that proximal third is the one that's going to be responsible for formation of the rough part of the right ventricle. Right? What we also need to know is the conus cordis. Right? So the conus cordis is going to form the smooth part of both ventricles. Which is this smooth part? We are talking about the outflow tract. Okay? We are referring to the infundibulum, and also we are referring to the aortic vestibule. Right? So these are the two smooth parts of the arch going to form. Right? From there, we have our truncus arteriosus. So this truncus arteriosus, so it's going to form two things, right? The pulmonary trunk and the ascending aorta. Right? So now we have talked about the bit of the embryology and the certain derivatives that are going to come from this. Right? So we now move on to the process of cardiac looping. Okay, so now we are done explaining the various parts of our heart. Now we want to know how this straight tube is going to form this complex heart structure. So this happens through a process that we refer to as cardiac looping. Right? So what happens is we realize that the bulbous cortex, if we look at these stages, right? We look at this diagram, right? We realize that the bulbous cortex and our primitive ventricle, they move down inferiorly, ventrally, and to the right. At the same time, primitive atrium in the sinus venosus, what it does is it moves dorsally, superiorly, and to the left. Good. Right. So what we are done with this kind of looping of the cardiac heart, we will form something like that. Okay. Just to name a few of the structures. Okay, we have this aortic arch, we have the pulmonary trunk. This is our bulbous cortex. If you realize now, it's moved to the right and it's moving inferiorly. And this is our left ventricle, which is our primitive ventricle. It's now inferior. And then we realize that the primitive left atrium is now moved from being inferior to being superior and more to the right. So this is a lateral view. Now, if we look anteriorly, you see, we are now forming that form of the heart that we all know. So from this, to familiarize ourselves, so we can now certainly say that this is the primitive right atrium, this is the primitive left atrium. This is the primitive right ventricle, this is the primitive left ventricle. And then this is the conus cordis. Very good. Okay. Now that we're done with cardiac looping, if you notice, we are referring to this as primitive right atrium, primitive left atrium. We have not yet formed the definitive right atrium and the definitive left atrium. So now, the stage at which we can now differentiate this completely is going to be explained through the process of septation, which is what we are going to be moving on to right now. Okay, so before we move on, you want to take it back through this diagram. When we mention that the sinus venosus is going to be incorporated into the right atrium, you want to notice this connection between the sinus venosus and the primitive atrium. So we are going to form the right venous valve and the left venous valve. But then, due to shunting of blood, we realize that the sinus venosus is moved more to the right, and it only has connection with the right atrium. So when this happens, we have it situated in the posterior superior aspect of the right atrium. So these two valves, right, dorsocranially, which is a fancy way of saying posterior superior, right, they fuse. So when these two valves they are fused, right, inside the right atrium, they form something that we refer to as the septum superior. Right. So they form, when they fuse, they form a ridge that we refer to as the septum superior. Right? This is, we need to take particular note of this because it's going to be helpful as we discuss septation of the interaction. Okay, so if we look at this is our atrium. So if we take a section, a transverse section through the atrium, we are going to notice something. If we take it like this, right? So right now we're in the atrium, we are looking into the ventricle. Understand that here outside is the atrium, inside is the ventricle. We have formation of endocardial cushions, right? So these endocardial cushions, they're going to form posteriorly and anteriorly. So these two endocardial cushions are going to come and then they're going to fuse. Right? So if we have noticed this pattern, so we have now formed these two canals, right? So these two canals, we refer to them atrioventricular foramen. We have formed our atrioventricular foramen by fusion of this anterior and posterior endocardial cushions. Right? So after this process, if we were then to take a section through the heart, a sagittal section, we are going to realize that we have got this cushion looks more like this. Right? So, and that means now this is the right atrioventricular septum. So this is the right atrioventricular canal, left atrioventricular canal. So now what happens during septation of the atria? We have, so now we are trying to septate the atrium. Right? So we have got these two. We have the formation of a septum, right, superior, and it moves and it approaches this endocardial cushion. So before it approaches, we have this connection between the primitive right atrium and the primitive left atrium. We refer to it as the ostium primum. In this septum, we refer to it as the septum primum. Right? So as this septum continues until it fuses with this endocardial cushion, it degenerates by apoptosis superior. So that means we have formation of a septum. We have formation of another canal joining the primitive right atrium and the primitive left atrium. And we refer to it, since it's the second connection, we refer to it as the ostium secundum. Right? We refer to it as the ostium secundum. So, before we form, we are now trying to cover this ostium secundum. Right? We have formation of another septum, which is thick. Moves it. Right? Right. So, as moves, you realize that it does not fuse together with the septum, right? No? It just sort of comes. So now at this stage is where we need to remember what we discussed. So imagine that we have our septum spirium in the right atrium. When we absorb the right wall of the sinus venosus, so this valve is initially large, right? So when we absorb the right wall into this right atrium, what happens is it pushes the septum spirium that it fuses with the septum secundum. When that septum spirium fuses with the septum secundum, we create now the foramen ovale, right? Or the oval foramen. So that means blood is going to be moving from the right atrium through the foramen ovale into the left atrium. Because initially in fetal life, we are saying that pressure is more concentrated on the right than on the left, right? So at this stage, we can now say that we have got a right atrium and a left atrium. So now we move on to the next stage, right? The next stage is formation of our interventricular septum, right? So we need to know that these ventricles, they are growing, right? The myocardial layer of the ventricles is growing, and the inner side is differentiating, the trabecular part is differentiating of our ventricles. As it's growing, we realize that there is fusion of as it's growing, we realize that there is fusion of our medial aspect of these two ventricles, right? So fusion of these aspects will form sort of this. Fusion of these aspects will form this muscular portion, which we are referring to as our muscular part of the interventricular septum, right? So this space between that muscular part of the interventricular septum and the endocardial cushion, we refer to it as the interventricular foramen. So we realize that while we are forming our aorticopulmonary septum or our conus septum, we realize that the space here, this space keeps on narrowing and narrowing, right? And also these endocardial cushions are also going to contribute again to formation of this septum that's going to sort of, this membrane that's going to come and fuse with this muscular part. And we refer to this newly formed area as the membranous part of our interventricular septum. So at this stage, we can now certainly say, number one, we have our right atrium, our left atrium, our right ventricle, our left ventricle. Right? So, but this is still not the structure of the human heart. We know that there is a valve here, there is a valve there. Right? So now we are talking about how we form those valves. If you notice, we have sort of this endocardial cushion. So this proliferation on the lateral aspects, right? So we realize that there is a proliferation of mesenchymal cells that exists around this region, right? It's here and it's like here, right? So what we need to know is that as blood is moving, blood is sort of narrowing that mesenchymal cells in such a way that this proliferation that was once here, it's going to just remain as tiny strips, right? These tiny strips that we are going to be forming, they are going to be joined to the muscular portion of this ventricle by small heart cords that we refer to as corda tendini, right? So we are saying, how are we forming our valves? We are saying that we have proliferations of mesenchymal cells, and then as blood is moving, it's sifting through this, we will form these narrow strips that we are going about, which are still connected to the muscles of the ventricle by cords that we refer to as the caudate tendon, and we refer to those muscles as our papillary muscles. Right? So, still on valves, we have the right atrioventricular valve and the left atrioventricular valve. The right atrioventricular valve, we refer to it as the tricuspid valve. The left atrioventricular valve, we call it the bicuspid valve or the mitral valve. Why mitral? So it comes from the word meter. Meter simply means the cap of a bishop. The cap of a bishop has two cusps. So if you remember that, if you have ever gone to a Catholic church, you know that the bishop wears a meter that has two cusps. That's why you refer to the bicuspid valve as the mitral valve. Very good. So now our heart is looking more like a heart. Right? But we still do not know how we are going to divide and form our aorta and our pulmonary trunk. Right. So now we are at this stage. So now that we have septated our ventricles, we still need to know how we are going to form our aorticopulmonary septum. So we have neural crest cells, right? So these neural crest cells, they are going to come into the region of the heart through the third, the fourth, and the sixth pharyngeal arc, and they're going to lie in the region of the outflow tract. Okay? So that's neural crystals that are found in this region. They are going to oversee or to stimulate the production of these aorticopulmonary septum. At the same time, they are the ones that are responsible. If you remember, when you mentioned formation of the secondary heart tube, those are the ones that are responsible for lengthening of that heart tube. Right? So we need to know three things, right? We have got formation of cushions, right? We form cushions, right, on the right superior wall, right, and on the left inferior wall. The ones on the right, they are going to move distally and to the left. The ones on the left, they are going to move distally and to the right, right? So such that they are going to spiral around each other. If you look at it, they are going to sort of spiral around each other. Say that if you are going to take a section through this section, and this section, you realize that there is different kind of orientation of this septum. You realize that initially it looks like this, then in the middle it looks like this, and then at the end it's going to look like this. So we need to know that there is that change of structure. Right? Because there is that formation of this spiral septum, this is the only reason why, number one, we have the aorta. Right? The aorta which is originating on the left ventricle. By the same time, we say it moves backwards, and then it moves anteriorly, and it moves to the right again, then moves to the left. All those gymnastics of the aorta and the pulmonary trunk, they are only possible because of this. And another thing that we need to know, that this secondary heart fluid that we were speaking about as being stimulated by our neural crystals, it exhibits something that we call laterality. So because it exhibits that laterality, we are able to form this. Because we are saying that the cells on the right, they are forming something on the left, the cells on the left, they are forming something on the right. So when we twist, it's able to form something like this. Right? Okay. Now that we are done with septation, we need to know that the aorta originates initially on the left side, it moves posteriorly, it moves to the right, it moves anteriorly. So I want us to notice the arrangement of our three vessels at three different sides. Right? So if we look at a section and the proximal aspect, we realize that the aorta begins from the left ventricle, the pulmonary trunk on the right ventricle. Right? If we look in the middle, we realize that the aorta is under or behind it, pulmonary trunk, and the pulmonary trunk is above, right? And then if you look at another portion above, we realize that the aorta is more to the right, the pulmonary trunk is more to the left, right? And then it curves, right? So basically the septation can be represented by drawing these points, and then sort of like this. So blood moves like that. The aorta, so you need to know that blood in the aorta starts to the right, from the right left ventricle, because behind the pulmonary trunk, it moves to the right, and then it carries there. There's the arc of the aorta. Very good. Now that we are done with septation, the next most pivotal thing is to talk about fetal circulation.